So this is an ADA enzyme. So first we'll look at the secondary structures which have been labeled according to color. So in red we have the alpha helices, there are 20 of those. We have the beta strands in yellow, and there are 8 of those. And in green we have the random coils. So all these secondary structures, along with the interactions between them, particularly the hydrophobic interactions at the core of the ADA enzyme, they all determine the tertiary and the secondary structure of the enzyme. So if we look for disulfide bonds, which contribute to the folding of the enzyme of the protein, if we label them as spheres, we can see that there are no additional spheres. There is only one sphere in the middle of the enzyme, in blue or cyan, and this represents the zinc molecule at the core of the enzyme. So its function is to maintain the stability and structure of the protein. Without it, it would lose both the structure and the stability, or there would be a change to the structure. So if we look at the eighth residue in the sequence, which is here, a lysine, and if we look at the 346 residue, which is here, it's also another lysine. Between these two lysines is the segment called the active, or the region called the active domain. So, right, let's see. So what happens when the ADA enzyme does not function properly? So in terms of RNA, adenosine cannot be broken down into inosine, and in terms of DNA, 2'-deoxyadenosine cannot be broken down into 2'-deoxyinosine. So why does this happen? It's due to a substitution in at the 247th nucleotide in the sequence, and this translates, the 847th nucleotide in the sequence. This translates to a substitution of an amino acid in the amino acid sequence of the ADA enzyme at the 282nd. Here we go, 282nd residue. 282nd residue, which is labeled in cyan on this. Here we go. This alpha helix, flagged by these magenta dots. So at this 282nd position, arginine is replaced with a glutamine. So it so happens that this substitution at the 282nd residue occurs within the active site of the ADA enzyme. So a hydrophilic arginine is replaced with a neutral glutamine. So this has implications to the active site of the protein, of the enzyme, because changing one amino acid for another with a different polarity and charge will affect the structure of the enzyme, and because this substitution occurs at the active site, the structure of the protein at the active site will change, and this will impact the ability of the substrate, either adenosine or 2'-deoxyadenosine, depending on if it's from RNA or derived from RNA or DNA, the substrate will not be able to bind properly to the active site, and therefore the 8 enzyme will not be able to catalyze the reaction of the substrate to the product. And that's why during the breakdown of DNA that there would be a buildup of 2'-deoxyadenosine when the 8 enzyme will not be able to function, and that will lead to 2'-deoxyadenosine interfering with DNA synthesis and repair, and can lead to severe combined immunodeficiency.